Good afternoon, everybody. On behalf of the board of Euro-Dyma, I'm very happy to welcome you to the third Farmers' Day of the association. At first, some news from Euro-Dyma. Since the last meeting, we have the two additional countries, Spain, with an association which is Bene in the northwest of Spain, and Serbia with Federation Norbs, which joined us this year. The geographic, the space perimeter of Euro-Dyma is enlarging right now. We have, of course, the European community, UK and Switzerland, and now adding some Eastern Europe countries. And we will move a little bit the status of the association to allow that. Also, we have a new member inside the Scientific Advisory Board, Dr. Stojan Peric from Belgrade in Serbia. And we have been very happy that Euro-Dyma has been selected to participate in the sympathetic program based on repurposing of drugs, a very huge program for the next 5 years funded by the European community. And that's the last one, but very important new. We are now opening a subgroup inside Euro-Dyma dedicated to clinical trials in Europe. It will be composed of some members of the scientific board, but it is not limited, and it will feature also some international experts. So if we look at the Scientific Advisory Board right now, Basil van Engelen, Denis Furling, Darren Monkton, Guillaume Basset, Benedict Schoser, Anne-Marie Ekström, Giovanni Meola, George Papadimas, and Stojan Peric from Belgrade, the youngest member of the scientific board. So if we go to the meeting right now, so the purpose of the meeting, as usual, as we did for the last two editions, is to share updates of ongoing studies and clinical trials from you, drug developers, targeting the European families and health professionals. Well, you know, the most advanced drug candidates are based on antisense oligonucleotide with various chemistries in order to penetrate deeply in the muscles. And so 4 pharma have accepted to participate in this edition: Artex, Dyne Therapeutics, Avidity, PepGen. And all drug candidates presented here are based on oligo. If we go to the meeting agenda, so we will start with Artex. The title of the talk is Development of a Lipid Conjugated Oligo to Treat DM1. And Judith will give the paper, will give the talk. Then Dyne will present the update and advance about their fourth platform to treat DM1, and Aaron Novak will do that. Then Avidity will come and will describe the current status of the Marina clinical trial with Li Yung Tai. And then we will finish through PepGen about their potential clinical trial, which will start probably this year. And that's it. And I now stop the sharing. And please, Judith, the floor is yours.
First of all, thank you to Alain and to the member associations of Euro-Dyma for inviting us to come and speak about what we're doing at Artex. Originally, it was going to be 2 of us, Beatrice and myself, Beatrice being our Chief Scientific Officer and co-founder, and myself, the Chief Medical Officer. Unfortunately, Beatrice is having some connection difficulty. She's actually on holiday and not in a place where she can connect easily, so you're stuck with me. But let's talk a little bit about what we're doing at Artex. And I have to say, we're very excited about what we're doing, and we're also very excited about what's happening overall in the industry. I think there's a lot of excitement in the space, and you'll hear from other companies as well. But I think what the key message for me is that there is hope here. I mean, there's a lot of different activities going on, so I really think this is a time to be hopeful, more so than at any other time in the development of approaches and to help patients with myotonic dystrophy and their families. So I think it's a very positive message. So let me talk to you a bit about Arthex. Some of you may know us, but we are a small company. We were initially founded by Beatrice, who is now our Chief Scientific Officer, and Ruben Artero, as a spin-off of a lab at the University of Valencia in Valencia, Spain. We have grown from that into a full-fledged biotech company, and we have a very seasoned and expert management team, including Frédéric Legros, who is our Chief Executive Officer and who's here on the call. And he has a long history of development in neuromuscular diseases and rare diseases. Myself as Chief Medical Officer, and Pedro, who is our Director of Operations. Now, Pedro and Beatrice are based in Spain, Fred's in Europe, and we are very much a European-based company. We do reach out to, and we plan to develop our lead compound ATX01 in myotonic dystrophy across the world, in fact, and we have a scientific advisory board that is composed of some names that you will recognize, and these are leading scientists, clinicians across the globe. So, as Alain said, we are, like the other companies you'll hear from today, are developing a therapeutic oligonucleotide. There's different types of oligonucleotides. Ours is an antisense oligonucleotide, which means that the sequence of nucleotides, the sequence of the little components, are complementary to an RNA that everybody already has in their cells. And in particular, our oligonucleotide is complementary to a RNA called miR-23B, and it is a class of RNA known as microRNA. So in fact, we are an anti-miR-23B antisense oligonucleotide, and the way it works is that we have carefully selected the sequence of nucleotides, these little elements that we want to be complementary to. We've modified them chemically, as Alain said, that it helps improve their resistance, helps get them into where they need to be, helps make them less immunogenic, less likely to cause immune reactions. And so as all the oligonucleotides have been chemically modified to some degree, but this makes it more effective, in fact. And it's not very long. It's only got 16 nucleotides. The nucleotides are the smallest component. And then what we do is we combine that with a lipid, a fat, essentially. And the fats are what, this particular fat is what helps the molecule, the oligonucleotide, get to where it needs to be. So when you combine the oligonucleotide with the lipid, you end up with what we call a conjugated anti-miR-23B, and that is what ATX01 is. We then dissolve this oligonucleotide into a solution called PBS, which is very commonly used, and it comes in a vial, and this vial is going to be injected or infused into patients. And probably we're going to start with every 2 weeks injections, but we might be able to go to longer intervals over time. And I don't need to tell this audience what myotonic dystrophy is, but what I wanted to point out on this particular slide is the fact that although many of the manifestations are muscular with weakness and wasting, myotonia, there are manifestations throughout the whole body. And this is important. And one of the things that we were very mindful of was to create a treatment that could treat all aspects of the disease, that could enter into different tissues. So we are very hopeful that we will be able to do that. And we have been able to show in animal studies that we can penetrate different tissues. So not only the skeletal muscle, the muscle in your limbs, but also the diaphragm to help you breathe, also the heart, which is a muscle. And we also penetrate the central nervous system in the animal studies. So we're very hopeful that we could have an effect not just on the muscular symptoms, but on other symptoms as well, which I think is important to the patient community. Now, how does our drug work? And I'm not going to try not to get too technical, but essentially, I think the take-home message here is that we have a dual mechanism of action. So we work on the MBNL protein. So you probably have heard a lot about MBNL protein is an essential protein to have, not only to have it in your cells, but to have it available so that it can do what it does best, which is regulate transcription of a lot of other proteins, a lot of other genes. And one of the things it does is it makes sure that a number of other genes, such as insulin receptor gene, genes involved in electrical conduction, genes involved in muscle strength, are working normally. So when your MBNL levels are low, or when your MBNL levels are not available because they're being sequestered or being held captive in the nucleus of the cells, then you will have problems with splicing of other genes, and those abnormal splicings will lead to symptoms. So what, and I think everyone on the call here knows this story, but what may be less known is the role that miR-23B plays, or microRNA 23B. So the interesting thing about microRNA 23B is that the longer your number of CTG repeats are, the more of it you have. And what this microRNA does, unfortunately, is it blocks production of MBNL. So not only do you have not only do you have MBNL being sequestered in the hairpin loops that are caused by the toxic mRNA in the nuclei, but its production is also reduced because of miR-23B. miR-23B also has a role in stabilizing these hairpin bends. So what happens, as you can imagine, is that you've got a double whammy. You've got reduced MBNL that's active and functional because either it's sequestered in the nuclei or it's not being produced normally. And that's not good. So what our approach has been is let's focus on the miR-23B. Let's inhibit it. Let's block the miR-23B. And so on the next slide, what we show is we show our product in the blue down below, and it binds with the miR-23B and it blocks it from working. And what that means is that now MBNL production can proceed normally. So we are producing MBNL, but also when there's no MBNL around, when there's no miR-23 around, what happens is that the toxic mRNA, DMPK mRNA, becomes unstable and it releases the MBNL. So you have both an increased production of MBNL and an increased release of MBNL from the foci. And what that does is it corrects the splicing defects and can improve the symptoms. So that's the theory behind how this works. And the question I'm sure you're going to ask next is, well, do you have any proof that it works this way? And on the next slide, I'm going to show you some of that proof. And we have, this is the top two graphs are from studies in human myotubes. These are grown from muscle cells from patients. And what we see here is that the amount of DMPK is reduced when we treat these cells with ATX01, our product. And we see that the MBNL protein increases, 3 to 4-fold, which is quite a lot when we add ATX01. We've also done studies in non-human primates, and we see also in non-human primates that when we give them ATX01, we reduce the amount of DMPK and we increase the MBNL protein levels. So we have non-clinical evidence to support this. In addition, we have done experiments in a very famous mouse model, the HSA-LR mouse, and this is a model that shows the muscle phenotype of DM1. And what we've done is we've done a number of experiments, but this particular one shows one where we injected these animals with doses every 2 weeks for 3 doses. And what we did, if you look at the bottom left graph, is we measured their grip strength. And what we found is that when you give the ATX01, the grip strength comes back to normal. So the colored dotted lines are showing you the different strengths of ATX01, and the purple area is what we would expect in a normal, unaffected mouse. Whereas if you just give placebo, an inactive substance, you see the strength is quite reduced, and that's with the gray line. So we're seeing that we can improve the strength in these animals back to normal. We also show in the graph to the right of that, that as we increase the dose, we also are showing improvement in the PSR, which is the percent splicing rescue. So the more we give, the more we get improvement in splicing rescue, up to 75, 80%. And then the other thing that we wanted to show you on the graph on the right is that we have the effect not only in muscle, but as you can see, what we're showing here are MBNL levels in different tissues in these mice. So we increase obviously the MBNL level in the muscle, but we also increase the MBNL level in the heart, the diaphragm, the brain. Less so in the kidney and the liver until we get to very high doses. And that's not a bad thing, because in the kidney and liver, they can cause, oligonucleotides can cause toxicity. So we sort of want to avoid the kidney and liver, and we want to make sure that the drug preferentially goes to the tissues where we need it to go to. And I think we have, at least in this mouse, this is what we're seeing here. So we're very hopeful that this will translate into the human. But of course, mice aren't humans. So we will need to do human studies, and we're getting ready to do that. In the meantime, we've done a whole slew of toxicology studies, and this is where we've given our drug to a number of different animal species to make sure that it's safe and well tolerated. And the bottom line is that, yeah, it is remarkably safe and well tolerated. We didn't see any what we call genotoxicity, where any genes are disturbed. We didn't see any immunotoxicity, where we created immune reactions. And we've got a therapeutic window that's quite wide, which means that we are able to increase the dose until we can see an effect. One of the difficulties when you're developing drugs is that you might get a dose that's effective, but at the level where it's effective, it causes side effects. So the really important thing is to make sure that your drug can have a really good beneficial effect at a dose that isn't going to create side effects. And so we've completed the non-clinical, and we're planning to go into the clinical study very, very soon. So I know this is what you want to hear about, so let me tell you about our first in human study. Now, this study is what we call a phase 1/2A study. It's double blind, which means that the subjects in the study will not know what drug they're on. Their doctors will not know. It's placebo controlled, so some patients will get active drug, some will get placebo. It is, and we're doing it in two parts. The first part is a single dose part, and then the second part is multiple doses. And we're doing this in patients with classic myotonic dystrophy type 1. Now, as you know, there are many, many different types of myotonic dystrophy. And we have to be careful when we do a study that we have a group that's fairly homogeneous, and that's hard in a disease like DM1 where there's such variability. But for this first study, we are going to be a little bit strict in terms of who can enter the study. Once we know that we have an effect, we will do other studies where we will broaden the entry criteria so more of you can participate. The main goal of the study, as with any first in human study, is going to be to make sure that our drug is safe. We know that it's safe in animals, but we need to make sure it's safe in humans. So we're going to start low and go slow. We're also going to look at what we call target engagement. Is the drug getting to where it needs to be and doing what it needs to do? And how do we check that? By doing muscle biopsies. I know that they're not pleasant things to have, but really for us to understand what's happening with the drug, we need to do muscle biopsies. And in those biopsies, we'll look at MBNL levels and we'll look at splicing index and see, do we see what we saw in the animal? The hope is that yes, we will. But we're also going to look at other things such as clinical endpoints. We're going to look at muscle function. We're going to look at the myotonia. We're going to look at patient-reported outcomes. And I think all of this is important to get a really good sense of, is this a drug that's going to be well tolerated? Is this a drug that's going to be effective? Is it worth pursuing further development? What dose do we need to take into the next study? Now, this study is called the Arthemir study, and you can see our logo up at the top, and we will be starting shortly. Just to get a little bit more into the study design, as I said, it's two parts, SAD and MAD. And in each part, we're going to have what we call cohorts. So a cohort is a group of patients. We're having 3 cohorts in the SAD, 3 in the MAD. Each cohort will have 8 participants. And in each cohort, 6 of those 8 will be on active drug, 2 on placebo. So patients who are participating have a 3 to 1 chance of getting an active drug. And the difference, the first cohort of the SAD is going to be at a low dose, a dose that is still efficacious in the animals at a low level. Then we're going to go to a slightly higher dose. If the first dose is safe, we're going to go to part 1, dose level 2, and then we're going to go to an even higher dose. If we need to, we might go to higher doses. But similarly in the MAD, we're going to start at a first dose level that's very, very low, and then we're going to go increase the doses. And this is what we call single ascending dose and multiple ascending dose. And the MAD will be like we did in the mouse study. We're giving 3 intravenous doses 2 weeks apart. Now, for each part, the patients will be followed up for 12 weeks afterwards, and then patients will have the option to enroll into a long-term extension study. So this is the overall design. It's not too different from some of the other studies that you may have seen. Now, as I mentioned earlier, we would love to enroll everybody in these trials, but we have to be somewhat selective, at least for our first study, to make sure that we don't enroll patients who are too sick, because we don't know how the drug is going to react in patients. We also don't want to have patients who are too well, because then we won't be able to see any improvement. So it's finding that balance, and these are the criteria we've decided upon. And again, there is an age limitation, so we want adults, but we don't want them over the age of 65 and over. We want a diagnosis of DM1, but not just more than 50 CTG repeats. We want one more than 150. And we want to have patients who have myotonia. And we're measuring this on something called the VHOT, which is video hand opening time. And we want patients who are not on any anti-myotonia medication. Patients who are on mexilotine or something else for the myotonia, after discussion with their doctor, may decide that they may want to stop that if it's not working, or but we need to have patients who have myotonia and are not on any medication for it. We are also going to be pretty strict, at least in the first study, on the cardiac disease. So because we don't know how this drug is going to react, and safeties are always our first priority, we want to make sure that participants have a pretty healthy heart. So we have some exclusions there, which means that maybe not everybody can participate in this first study. However, if things look good, and we didn't see any problems with the heart with our drug in animals, but you never know. But if we don't see any problems in this first study, later on we will make the drug available to patients who have more cardiac disease in future studies. And in terms of what we're going to measure, we've talked about this a little bit. I think most of you will be very familiar with these outcome measures. Of course, the priority is safety and tolerability. So we're going to do some lab work. We're going to do some cardiograms. We're going to ask the participants about any adverse events they're having. We're going to do blood and urine levels to measure the level of our drug. That's called pharmacokinetics. And we're going to do pharmacodynamics, which means looking at how the drug works in the body, and we're doing that through muscle biopsies. And then clinical testing, we're doing the muscle testing, and of course some of you may be familiar with a lot of these different measures. They're fairly standard. And we're also going to look at a few other things, because we think we might have nervous system effects, brain effects. We're going to look at an apathy evaluation scale. We're going to look at a fatigue and daytime sleepiness scale, and a few other patient reported outcomes to really get as best an idea as we can of how the drug's working, even in this very preliminary small study. So when are we doing this study? Well, we are on track in the US to file an IND in the third quarter of this year. Europe will follow shortly after. We have the European regulatory authorities, the EMA, has asked us for a little bit more stability testing on the drug, just to make sure that its shelf life is a little bit longer. And so we're going to do that, which means the US will start the study first, but we are definitely going to be coming to Europe, and we'll definitely have some sites in Europe. We're expecting the first patient to be enrolled in the fourth quarter, at least in the US. Europe, it might be January 24. We'll see how that goes. But even by the end of 2024, we expect to have some early results. And the study will be done in the US, Europe, in Canada, and possibly also the UK. So we're currently in the midst of speaking with some potential investigators and seeing if they're appropriate for participation in this study. And finally, I wanted to talk about some other things we're doing, because based on the mechanism of action of ATX01, we could have an effect not only in type 1 DM, but also type 2. And I don't know if there are any type 2 patients or families on the call, but this is an area that we're quite interested in, and our drug might work in that as well. So if we see effects in the first study, we would likely do a study in type 2. We also are keen to do a study in congenital DM1. I mentioned earlier that in the first study, we're not enrolling children, but if we see promise in the first study, then it would make sense for us to look at congenital DM1. And we also have some other products in the pipeline as well. So we are building a biotech company based on the treatments using anti-MERS, which is a very exciting new area of research. And I was going to end here. And again, I want to come back to this message of hope. You know, at Arthex, we really believe that things are changing. We saw some great progress to date. And I think there's lots of different people looking at lots of different angles to try to make a difference. And ultimately, that is our hope, that we make a difference to the lives of patients and families. Thank you so much for listening.
Thank you so much, Judith. It's a kind of talk that we would like to hear every week. Very exciting. So it appears that in the first trial, you don't expect to have children. So it will come later. I have understood that. When do you expect congenital form? And do you plan to test infantile form?
Short answer is yes, eventually. I'm going to be a bit vague here, because I think what we need to do is we need to see the results of this first study. Then what we need to do, and one of the things that I think as a community that we're looking at is there's not as much known about congenital infantile DM1 as there is about the adult classic form. So one of the things that we are keen to do is to work with those of you who are in this space to understand a little bit better the natural history. You know, we have a little better handle on the natural history in adults, and I think to progress clinical trials, particularly in rare diseases, it's critical that we understand the natural history. So this is an area that we're starting to look at: is what is the natural history? What is happening in babies and children with DM1? We also need to start thinking a little bit more about outcome measures, because we're not going to use the same outcome measures in children necessarily that we use in adults. We may not use, some things, babies can't do that adults can do. So we need to start paying a little bit more attention to that. And I'll be honest, that will take some time for us to develop that. But it's certainly, obviously an area of great unmet need that we are very keen to tackle.
Okay, thank you so much. Any question in the small audience of this meeting?
No, it's a very interesting story. I have one question. I was just wondering, during your presentation, you said it will also improve the symptoms. In other words, so the problems will become less when it all works. Is that right?
That's the hope. The hope is that this should lead to improvement in symptoms and slowing of the disease. That is the hope.
Yeah, I had the impression that it's only slowing the disease, so stop the disease, but it's really promising. Where you hope for?
Yeah. No, I think, for instance, and we've seen this with the Avidity study, when you improve splicing of the chloride channel, you improve the myotonia. So again, I think any of these treatments that are going to improve the splicing will improve the splicing of the chloride channel, will improve myotonia. The degree of improvement in other symptoms we'll need to see. There may be a point where if too much damage is done, there's no turning back, but I'm very hopeful. And we've seen this in with the avidity data, that once the splicing is corrected, there can be definite improvements in symptoms. So we are hopeful.
Nice. Yep. That was my only question.
Okay.
Thank you.
Thank you so much. Well, I think we are almost on time. Thank you very much, Judith. And now the floor is for Aaron for Dyne Therapeutics.
All right. So thank you, Judith, for that great presentation. Covered a lot of stuff I was going to cover, so I'll go a little bit faster through some of those items. But thank you very much and good afternoon. My name is Aaron Novak. I'm an executive director in the medical affairs group at Dyne Therapeutics. I'd just like to take a second to thank Euro-Dyma for allowing us the opportunity to speak today. It's always a great honor to speak to the DM community, especially today about the progress of our clinical trial, specifically Dyne 101, which has been designed to treat adults living with myotonic dystrophy type 1. Next slide. These are our forward-looking statements. You can refer to the website if you're interested in more of the detail. It's very exciting. So we go to the next slide. So at Dyne, it all really starts with our mission. We are laser focused on delivering life-transforming therapies to patients living with muscle diseases, with DM1 being our priority. Our mission is core to who we are and what we do every single day. We're incredibly excited to inform you that we have started dosing participants in our Dyne 101 clinical trial, which is called ACHIEVE. And our focus today is sharing an update on the clinical trial progress, as well as details about the clinical trial of Dyne 101. Go to the next slide, please. So we are building at Dyne a leading muscle disease company to deliver for families living with DM1 and other muscle diseases. Shown here are really our three core strengths: so delivering for patients, DMY expertise, and Dyne 101. So I'll start first with the commitment to delivering for patients. We've initiated our clinical development program in DM1, which has been informed by the patient community and the scientific leaders in the field. Second, you'll see here we have exceptional in-house advisory expertise with a deep knowledge and years of experience in DM1, as well as developing novel antisense oligonucleotide therapeutics. We also have a world-class scientific advisory board, which includes luminaries such as Charles Thornton, known to many people on the call. And third, definitely not last, Dyne 101, which is our clinical candidate that is designed to reduce toxic nuclear DMPK and correct the splicing defects to potentially stop or reverse the disease progression. We intentionally designed Dyne 101 to overcome one of the biggest limitations, which is known for antisense oligonucleotide therapeutics, which is really effectively in delivering these modern oligonucleotide therapeutics to muscle tissue. So next slide. I think Judith went through a lot of this, and I think everyone on the call is very well aware of this. But just touch on a couple of things that I think Judith also touched on was really the multi-systemic nature of DM1, and the fact that it not only leads to skeletal muscle weakness, myotonia, and other weakness in skeletal muscle, but also some of those CNS-related issues with fatigue, cognitive deficits, daytime sleepiness, but also with respiratory, gastrointestinal, and importantly, cardiac issues. And one of our tenets here at Dyne is really to focus on treating all of those symptoms, and our therapies, as you'll see in a little bit, have been developed to target all of those issues, from the skeletal muscle, cardiac muscle, smooth muscle, and GI, as well as the CNS part of the body. We know that all these symptoms greatly impact quality of life of people living with DM1 as well as their families. And again, we are dedicated to approaching and attacking all of these different symptoms. DM1 is believed to affect around 1 in 2,300 people globally. And although we recognize it is a rare disease, DM1 is not so rare and often impacts multiple generations within a family. So go to the next slide. Okay, so just briefly, again, we'll just touch quickly on kind of how proteins are made. I think it will be helpful to understand, again, as Judith went through, some of the mechanisms of action of the disease, as well as how our product works. So again, this slide describes how proteins are made. It starts with cells. You can see there a picture of the cell with the nucleus and the cytoplasm. You have the DNA inside of the nucleus. Now again, the DNA is a substance that lives inside the nucleus and contains instructions for making these eventual proteins. A section of the DNA, so I'll pick one of those little red parts, for example, is what's called a gene. The DNA information on those genes is then written into an RNA, which is called transcription. The RNA is then cut or spliced into what is called messenger RNA, or mRNA. And then the mRNA is exported from the cell, where it's then translated into proteins. So again, this gives you a little sense for kind of what happens in a normal process. We go to the next slide. I won't go into great detail because I think many of you guys know this, and as well as Judith captured a lot of this. But again, hopefully it's a useful reference. So again, briefly, this slide, what it shows is that the defect in DMPK gene is what causes DM1. It is well known that DM1 is caused by a high number of CTG repeats in the DNA section of the DMPK gene. And specifically, people with DM1 have abnormally high levels of these CTG repeats in the blood and the tissue. What happens is that these CTG repeats are copied to make the mRNA. After splicing the abnormally long CTG repeats, toxic mRNA that traps the important proteins occurs. You can see here these hairpin loops, which again attract and trap these MBNL proteins, which have a function have a function to regulate and process other splicing events and other mRNA inside the nucleus. So again, these trapped proteins form clumps, which are called foci. And these proteins then can't leave the cell, and these lead to abnormal splicing. And the abnormal splicing is what's believed to cause many of the symptoms in DM1. So as you can see, to reduce or knock down this toxic DMPK mRNA, any potential therapeutic would need to get into the cell and into the nucleus to be able to work. So next slide. Again, just I think it's well known to everyone that currently there are no disease-modifying treatments available for DM1, and currently treatments that are available aren't really focused on managing the symptoms of the disease and not necessarily the genetic cause of the disease. So to treat the genetic basis of the disease, key is to get a therapeutic agent into the nucleus of the cells and knock down the nuclear toxic DMPK RNA and correct the splicing effect, which may lead to slowing of the disease, or stopping the progression. Prior attempts to treat prior attempts to create therapeutics targeting muscle disease have been limited by the poor delivery to muscle cells. Now, what we attempt to do is through our targeted delivery approach. It's designed to effectively and efficiently deliver a therapeutic agent to muscles of patients so that we can make a meaningful impact on their lives. As I go through some of our preclinical data in a few minutes, how our investigational medicine will become a bit more clear. All right, so this next slide, I'm just going to spend a few minutes on because I think it's an important slide, really to focus and kind of describe the FORCE platform and DYNE-101. So the FORCE platform really drives all of our efforts to develop and deliver muscle-targeted therapeutics. It is expressly designed, as I mentioned earlier, to address one of the major issues in muscle disease, which is efficiently and reliably delivering antisense oligonucleotides, or ASOs, on this slide, that target the genetic basis of the disease in skeletal, cardiac, smooth, and CNS muscle. As you can see, Dyne's FORCE platform consists of three parts: an antisense binding fragment that we call a Fab to enable the targeted delivery of the ASO to the muscle, a clinically validated linker, and an ASO selected to target the genetic basis of the disease. I'll start by describing these three components, and then I'll kind of circle back to the importance of targeting the transferrin receptor 1 to drive this targeted delivery of our investigational medicine. Now, in this case, we chose a monoclonal antibody. We chose a portion of the monoclonal antibody, which is what we call the Fab. So it's just a fragment of that full monoclonal antibody. We believe that this may offer a powerful advantage over the larger monoclonal antibodies, including the fact that it's a smaller size, has a lower protein load, and has the potential for an enhanced tolerability effect. Again, which we'll have to see how that plays out in the clinic, but that is what it was designed to do. The goal is ultimately to enable targeted, effective delivery of the therapeutic molecule to, as I mentioned before, skeletal, cardiac, and smooth muscle. Have a look at the linker portion. We selected what's called the val-cit linker, so valine citrulline, for our platform. We chose this based on the fact that it has a serum stability that is known and quite well. It's also clinically validated both for safety and efficacy, and some already approved anti-drug conjugates that are on the market. And we believe this further de-risks our FORCE platform. So if you look at the Fab and the linker, those two portions, those two parts of the molecule stay consistent across all of our programs. And what changes is the third component, which is in this case the ASO. And in the case of DM1, this was chosen to target the genetic basis of DM1. We have other programs for DMD and FSHD, and those different ASOs are different in those cases, again, to target the specific genetic basis of those diseases. So what we think is the versatility and modularity of our scientific platform provides flexibility to deploy different types of ASOs and other agents that are rationally selected to match the target biology of the disease. For disease-modifying nuclear knockdown is key, which is the case for DM1. It is critical that we select an ASO that is preferentially trafficked to the nucleus. For this reason, we designed an ASO that targets the toxic DMPK specifically in the nucleus. Now, over on the right here, you can see kind of how the DYNE-101 works and how it again binds the TFR1 receptor and then is brought into the cell and into the nucleus. And again, we think it's different from some of the other approaches that are currently being studied. What we're doing is really essentially using the cell's natural biology to deliver the therapeutic to the muscle cell. Our Fab binds tightly to something called the transferrin receptor type 1, or TFR1. The TFR1 receptor are highly expressed across the surface of muscle cells, including skeletal, cardiac, and smooth muscle. In normal cases, the TFR1 receptor is known to participate in iron homeostasis. So we're able to, again, to harness this already existing mechanism on the surface of cells, deliver our therapeutic with this receptor-mediated mechanism. And by targeting the TFR1 with DYNE-101, we hope to have the potential for wide safety margin, sustained effect, which again, hopefully will result in less frequent dosing and chronic administration. So hopefully this gives you a little bit of sense of the FORCE platform. And again, for DYNE-101, with the ASO being specifically target the toxic nuclear DMPK. All right, so the next slide. Okay, so as we prepared for the clinic, we did a number of non-clinical studies. And the next few slides will really highlight those critical studies and some of the key data that we generated before going into the clinical trial. Next slide. Okay, so to prepare for entry into the clinic, Dyne has deliberately conducted comprehensive preclinical studies across our programs. And we have not shied away from asking the tough questions or running the right studies to evaluate our product candidates. We've operated with a strong desire to understand how our molecules best position for success in all patients. The team has generated the most comprehensive, significant, and robust data set across our programs, and we believe sets a new bar in the field, whether it be with in vitro cell lines, various mouse models. So we'll talk a little bit about the DMSXL mouse, which have an HSLA mouse data. But also, we also looked into non-human primate studies, and all of this is really to look into see the pharmacokinetics and pharmacodynamics in both diseased muscle models as well as some non-disease models, non-human primates, and to all understand how this force is distributed to the muscle and how we can see some safety and efficacy. I do want to point out just for 1 second the importance of the HTFR1 DMSXL mouse model. So literature indicates, we've been talking about, that the toxic human DMPK RNA, that is the genetic basis of DM1, resides within the nucleus. So we really wanted to set out and conduct our own experiments to understand this further, as well as the ability of DYNE-101 to reduce this toxic nuclear DMPK. We developed the HTFR1 DMSXL mouse model, shown here, by crossing the human TFR1 mouse with a DMSXL mouse that carries the mutant DMPK transgene with over 1,000 CTG repeats. So by crossing and by getting the human HTFR1 receptor, as well as the DMSXL human nuclear toxic DMPK, we're able to test our actual clinical candidate, DYNE-101, and we don't have to use a probe molecule or a tool molecule. So again, this animal model has given the potential to increase the translatability to patients who also accumulate toxic nucleic DMPK. So the next slide just is one quick little bit of a slide on some of the data that we have within the TFR1 DMSXL mouse model. In this case, we evaluated DYNE-101 in the heart of a homozygous mouse. We did 2 doses, a relatively low dose of 10 milligrams per kilogram on day 0 and day 7. And you can see here on the left, compared to the saline-treated animals, that we see a robust reduction of 49% of the toxic DMPK. In the middle, you can see again, compared to the saline-treated animals, which you can see that in red, kind of pink in the blue cells, the foci. And then again, after treatment with these 2 doses, you can see that there was again a 49% reduction of the area of these nuclei and foci within the nucleus. And all of this, reduction of DMPK and reduction of the foci leads to, again, you can see here, robust splicing correction. You can see here in the middle, in the black, which is the mouse treated with saline. And again, after 2 doses of DYNE-101, you can see a reduction of the splicing and correction of the splicing in these animals. So again, I think we chose the cardiac muscle because historically, the cardiac muscle has been difficult to assess in these muscle diseases. And we know that in DM1, it's critical to improving the condition for patients. I'm not showing all the data, but we have seen similar results of DMPK knockdown and splicing correction in other important muscles and cell types, including skeletal, respiratory, smooth muscle, and recently presented the CNS. So again, we are very excited about this data from the numerous studies in the HTFR1 DMSXL mouse. And we believe that working with this model really sets a new standard for evaluating DMPK knockdown and splicing in DM1. So the next slide is just a bit of a summary. So in addition to these mouse models, we are also utilizing non-human primates to evaluate the ability to get into muscles and engage the target, which in this case is DMPK, to assess the safety and tolerability in these larger animal models. It is important to note, again, just want to remind people that monkeys are not a disease model, as they are healthy. And so what we're looking at is knockdown of wild type DMPK. In our non-human primate studies, we demonstrated that DYNE-101 robustly reduces the wild type DMPK in skeletal, cardiac, and smooth muscle with up to 70% knockdown. Again, this data demonstrates that we can deliver and engage the target of these large animals' muscle tissue. We also evaluated in the non-human primates a 13-week GLP toxicology study. As shown here, we are very pleased that DYNE-101 has a safe and well-tolerated profile after repeat dosing. We observed no dose-limiting toxicity and no changes in cardiac, respiratory, liver, or kidney function. All right, so next slide. Okay, now as we prepare to go into the clinic, we have done extensive work to prepare to advance DYNE-101 to the clinic. We've engaged with stakeholders around the globe as part of this effort. We've engaged many people in the advocacy community, patients, as well as key opinion leaders from various disciplines, including neurology, cardiology, physical therapy, pulmonology, as well as other areas. So we get a crucial perspective on how to inform our efforts. These thought leaders, with their tremendous experience as clinicians in running clinical trials, have provided us with the critical input on the overall design of our clinical trial. We'll be studying biomarkers such as splicing, as well as myotonia, various strength and functional outcomes, as well as key safety considerations. And what we're lucky to do is leverage insights from the NDM1 natural history study. Again, we use this to inform us on the design of our clinical trial program. And the ability to work with this study provides great insights for our own program, as well as informing on biomarker testing methods and endpoint selection, as well as providing access in the future to study data, as well as biological samples going forward. We've also reached out broadly to the advocacy leaders, patients, and caregivers, many of them who are healthcare providers themselves, to ensure we have a patient-centric study design and to provide advice in a number of areas, including clinical trial assessments, placebo and biopsy-related considerations, as well as clinic visits and the design and the decision-making insights for the selection of participating clinical trial sites. So there's a little bit of background on what we did as we prepared to go into this, the ACHIEVE study. Go to the next slide. Okay, so this gives you a little bit of information about our clinical trial. So our global clinical trial for DYNE-101, which is called the ACHIEVE study, is a randomized placebo-controlled trial consisting of essentially 4 periods. We have a 6-week screening period, you can see on the left, followed by a 24-week placebo-controlled multiple ascending dose period. We have 4 different dose levels. Judith described what a multiple ascending dose study looks like. So we're studying 4 different ascending doses, where individuals will receive either placebo or DYNE-101, and we're administering it either every 4 weeks or 8 weeks in the different cohorts. This is followed by a 24-week treatment period where all participants will receive DYNE-101 administered every 4 to 8 weeks. So if a patient was on placebo in the multiple ascending dose portion of the study, they will then transition in the treatment portion of the study to DYNE-101. So everybody will receive active treatment after 24 weeks. This is then followed finally by a 96-week long-term extension period, where patients again are just followed for safety and efficacy endpoints for an additional couple of years. So it's important. I just want to again highlight that it's important to note that these patients who receive placebo in the multiple ascending dose portion will eventually transition and will be given an active treatment after 24 weeks in the open label extension and the long-term extension. And some of the endpoints that we hope to study in this trial are safety and tolerability. We're looking at splicing data, pharmacokinetics, and pharmacodynamic information, again, to understand how the body is processing DYNE-101 and how it is acting in the body. We'll also be looking at various measures of muscle function and strength. And this also helps us understand how the body is functioning. And we're also carefully assessing lung and heart function in the trial. Again, with the help of our expert advisors and our PIs in the study, we are confident that our study design is robust, will measure key outcomes, and is striving to manage the burden on families and take into account the assessments that aren't too burdensome for the participants themselves or the families that look after them. And we're excited to say that we will be communicating some safety, tolerability, and splicing data in the second half of this year. So we'll be happy to come back and talk once we have that data out into the world. So if you look at the next slide, it gives a little bit of information about some key inclusion and exclusion criteria that must be met for a patient to participate in our study. So if you look here, that we're enrolling around 72 men or women. They have to be between the ages of 18 and 49 years of age. They have to have a DM1 diagnosis with a CTG repeat size more than 100. The onset of muscle symptoms have to have taken place at least by the age of 12 years of age. And this kind of gives you that adult, or people call the classic form of DM1. Hand opening time, based on the physician's assessment, has to be at least 2 seconds. We'll also be measuring hand grip strength and ankle dorsiflexion at the inclusion criteria, and people have to meet a certain criteria to get into the study. Again, as Judith kind of described, we want someone who's not so affected that they won't see a benefit, but also want people who are so unaffected they also won't see a benefit. So we're trying to get that. This is a first-in-human study. We're trying to get that right population so we can see an impact. Participants won't be able to use assistive devices such as canes or walkers. And there are a number of additional inclusion-exclusion criteria, as well as a whole listing of all of our primary and secondary endpoints on the clinicaltrials.gov website, as well as the UDRAC-CT website. So you can kind of see it on the bottom. It's a little bit small. I'm happy to send it to Alon afterwards to help people if they're interested in finding those postings. And again, just wanted to highlight that in addition to finding the inclusion-exclusion criteria and the additional endpoints, you'll find that all the studies, sites, and countries, as well as the PIs and their contact information on these postings. And so currently, the study is open and enrolling patients in New Zealand, in France, in the Netherlands, United Kingdom, Italy, and Germany. And so again, you can see on the ct.gov the individual sites within those countries, as well as the primary investigator and the contact information if you're interested. So the last slide. again, I just wanted to say thank you, everyone here. We can't do our work without your support and the encouragement and the sacrifice of the community. We understand that there are countless dedicated scientists, physicians, physical therapists, and other healthcare providers around the world that are needed to treat myotonic dystrophy, as it is a devastating disease with no approved treatment options, especially for people like Suzette and Billy Dean, shown here on the slide. And again, I wanted to mirror what Judith was saying about the optimism. I think it's really amazing how many companies are working in the space and the different approaches. And I think it's a really exciting time for DM1 and for people living with DM1 and their families. So thank you for the opportunity to talk to you guys today, and I'm happy to answer any questions that you might have.
Thank you so much, Howard. Very nice, very interesting, very impressive talk. Just same question as for Judith at the previous talk. Do you plan to have children in your cohorts in the next trials? Not for this one, because you have limited to 18, but do you expect to enlarge the range of ages?
So I'm gonna give you a similar answer, Judith. Is yes, we are. The short answer is yes. We are definitely looking into that. We're looking at expanding our clinical development program. As you saw with the inclusion exclusion criteria, there are patients who will be, or participants who will be excluded based on some of the muscle functions. We also have, which you didn't mention, some cardiac inclusion exclusion criteria. So there are no people who will be excluded in addition to children. And so it is something we're looking at, because our goal is to really impact all the patients. And so we're looking at expanding in future trials, similar to what Judith was mentioning. I think we're learning a lot about how to assess children with congenital and pediatric onset DM1, because again, some of these endpoints are not well studied. I think we'll learn a lot from the AMO study as it starts to read out data. And we're spending a lot of time talking to experts, expert neurologists, and looking into other fields who do study some of these, for example, daytime sleepiness and some of these cognitive deficits. So it's a long-winded answer, really, to say yes, we are looking. We will add them in the future. The timing is not yet known. I think it's really important that we get this proof of concept in the first in-human study in the adults, and then we'll pivot from there.
Okay, thank you. And did you already deliver doses in patients in one of the sites?
Yeah, all of our sites are enrolled.
So it started?
Well started. Our first patient was dosed, I think, late last year.
Okay, good.
Yeah, all the studies are enrolled.
Any questions from the audience?
Yes, I have a short question. Sorry, I just jumped to the presentation, but very nice result. Any idea about the way, or if the product is able to cross the blood-brain barrier and go in the brain?
So, I mean, it is well known that the blood-brain barrier has and expresses TFR1. There are a number of other companies that are actually using TFR1 for specifically neurological disorders. One company called Denali, for example, they're actually using TFR1 antibodies to cross the blood-brain barrier. So, again, it is well known that it does cross the blood-brain barrier. Again, we are interested in kind of affecting the multisystemic nature of the disease, including the CNS symptoms. And I didn't show the data really, but recently at the ASGCT meeting in Los Angeles a couple months ago, we did show that in both monkeys and this mouse model that we're able to cross the blood-brain barrier and get significant uptake into the cortex, the deep brain, as well as the cerebellum. And again, we saw some really interesting data on knockdown and reduction, knockdown of the DMPK RNA in the cerebellum and the neurons, as well as foci reduction. So again, I think that data really did tell us that it's possible to cross the blood-brain barrier. And we saw really significant distribution throughout the brain. So hopefully that answers your question.
Thank you. And Giovanni has a question.
Thank you for your talk. I have one question. Giovanni Meola from Milan, Italy. What about patients with problems with the heart? I mean, patients who have a pacemaker or ICD defibrillator. Are those patients excluded in the enrollment or could be enrolled? Thank you.
Yep, of course. From the current study, they are excluded, so they're not able to come into the current study. But as I was saying earlier, we are looking to expand, in future studies, the inclusion exclusion criteria to try to, capture all of those participants who might be excluded in this trial. I think everybody would understand, and I think Judith went through it really well, was that we really want to be, in this case, get a homogeneous population to improve our chances of seeing an improvement and then getting this to patients as soon as possible. So it's definitely the plan in the future, but in this current trial, they are excluded.
Okay, thank you.
Peter?
Hello, thanks. Interesting presentation, thank you. I was just wondering, I believe you were looking for 72 participants. I was just wondering how recruitment was going and if we as patient organizations can do anything in particular to help if needs be.
Yeah, so maybe I'll start from the last part. I think we're always looking for help. I think it would be great if the patient community, the patient advocacy groups could help. I know a number of the physicians in the countries where the study is going on have trying to work through them. So again, I think going to the clinicaltrials.gov or the UDRAC CT and seeing the countries and maybe perhaps reaching out to them and seeing how you could help bring awareness to the community about the sites that are open. I can't get the specifics of enrollment per se, but I can just tell you it is going quite well, and we're enrolling. We're pleased with the enrollment that we're seeing. Hopefully I answered your question.
Thank you.
Thank you.
Okay, thank you so much. I think we'll move on, and for Li Yongtai from Avidity right now.
Thank you so much for the opportunity to present to you. My name is Li Tai, and I'm the Executive Director of Clinical Development and lead of our myotonic dystrophy program with the drug AOC 1001 here at Avidity Biosciences. I first want to thank you, Euro-Dyma, for inviting our team to share about Avidity and our progress in developing potential treatment for the myotonic dystrophy community. We view this as an important opportunity to continue our dialogue with you and look forward to your feedback. So this next slide is a forward-looking statement during today's presentation. So Avidity is a biotechnology company based in La Jolla in San Diego County in United States. At Avidity, patients are the heart of everything we do. Our mission is to improve the lives of people affected by diseases with limited therapeutic option by advancing a new class of drugs. And our team has extensive experience in drug development, and also we're committed to involve patients and families at every step of the way. And this is the foundation of our company and is really what drives us in advancing new treatments. As you know, as Aaron did such a good job explaining or just summarizing rather DM1 disease and disease mechanism, I'll go through this quickly. As you know, DM1 is caused by a mutation in the gene DMPK, and with hundreds of thousands of nucleotide repeats that build up at the end of the DMPK gene, leading to the pathology of the disease. And the ultimate result of this toxicity is a DM1 disease signs and symptoms that really affect multiple organs in the body. Specifically, that's important for AOC 1001 based on its mechanism. A substantial amount of this disease signs and symptoms are in the muscle tissue, including skeletal, smooth, and heart muscle. And these symptoms are well known to you, so I won't belabor, but we are really focusing on the muscle and heart tissue to reduce the amount of toxic DMPK mRNA made by the cells, and we hope that by doing so, we can decrease some of these signs and symptoms. I'm going to tell you a little bit about our technology. Our drug platform really unlocks a new way to deliver drugs directly to affected cells like the muscle and heart. And this allows us to develop new treatment for diseases with currently limited therapeutic option. Our potential treatment is called antibody oligonucleotide conjugate, or AOC for short. We're applying our technology to develop AOC 1001 for the DM1 community, which has the potential to address the root cause of the disease. The AOC platform it really has three components as well. It combines the proven technology of existing drug types that are well understood for each of their specific properties in drug development. We have an antibody, which is a naturally occurring protein that's used to fight infection. And it's commonly used in drug development because they can be made to target almost any protein, including those that are different between one cell type or another. And antibodies are foundational for over 100 approved drugs, many of which are for chronic conditions. So it has a proven profile of safety and efficacy. In our situation, we're using the antibody as a vehicle to deliver our drugs to muscles and heart cells. And this antibody is linked by a linker to an oligonucleotide. It's an oligos short strand of DNA or RNA. And it can be a powerful drug because they can change gene expression, which then affects how a protein is made downstream. There are multiple approved oligonucleotide drugs, and many of which are disease-modifying treatments. So in the case of AOC 1001, the type of oligo we're using is called small interfering RNA or siRNA. It works by harnessing a natural biologic process in the cell called RNA interference. And this is a mechanism that's used by the bodies to fight off things like RNA-containing viruses. So what happened is that siRNA is loaded into a protein complex shown here as a Pac-Man. Once loaded into the protein complex, it will chew up a target RNA that binds to this siRNA. In our case, this target RNA will be the DMPK RNA. So this slide puts everything together and shows how AOC 1001 works. The antibody we're using sticks to a certain receptor, which is the transferrin receptor 1, and which leads to the entry of this drug into the cells. And then once inside the cells, the oligonucleotide is released, and then it can go to the nucleus and in general to bind to the protein complex, that's the Pac-Man, and then chew up the toxic DMPK RNA. So I'm excited to give you an update of the MARINA trial. I was reviewing what we presented last year, and last year we really talked about the design, and now it's really gratifying to be able to share some of the results with you. Before we get into the data for MARINA, it's very important to acknowledge the important contribution that has helped us get to where we are today. We're really leveraging the important progress made by developing a network of advocates, experts, using knowledge and tools, including the Myotonic Dystrophy Clinical Research Network, the NDN1 Natural History Study, our patient advocacy partners, including Euro-Dyma and DN1 community of patients, family, and caregivers. And we also want to thank especially the participants of the MARINA trial, and their families, and as well as the sites, investigators, and their teams for their time, commitment, and continued contribution in the MARINA and the MARINA OLE open label extension study. So before I go into the overview of the program, I want to share a quick update of the program. As we have disclosed previously, the MARINA clinical trial is on partial clinical hold since last September by the FDA. This is in response to a single serious adverse event that occurred in a single participant at the 4 milligram per kilogram, the highest dose tested. The event was a bilateral ischemia in the region of lateral genicular nuclei in the thalamus with subsequent hemorrhagic transformation. We have conducted an extensive investigation into this event, and as we disclosed, we have not been able to find any plausible biological link to any components of the AOC 1001 or the AOC platform, or to the transferrin receptor mediated delivery mechanism or the reduction in DMPK mRNA. We're continuing to partner with the FDA, and we will share information as soon as we have them. But the MARINA trial in general, it's completed with 38 patients enrolled, and we are also remain on track to provide you with a open label extension study data at the end of the year. So now a little bit about the design of the MARINA trial. The three main goals of the MARINA study are to evaluate the safety and tolerability of AOC 1001, to understand the pharmacokinetics, which is how the body is processing and breaking down the drug, and also to evaluate the ability of AOC 1001 to reduce DMPK mRNA in the muscle biopsy. There are two parts to the study, Part A and Part B. In Part A, participants receive a single intravenous dose of either 1 milligram per kilogram of drug of AOC 1001 or placebo, and then were followed for 6 months. In Part B, participants receive either 3 intravenous doses of either 2 or 4 milligram or placebo, and then follow for 3 months. So the total duration is also a 6-month trial. After this follow-up, participants can elect to enter the extension trial. And to date, 37 of the 38 participants in MARINA trial have enrolled into MARINA open label extension. Now we've got quite a lot of clinical data across both trials with over 132 doses of AOC 1001 given, and also over 27 patient years of follow-up. This is the baseline demographics of the study. You can see across the top there, the placebo 1, 2, and 4 milligram dose cohorts are listed by columns. And one key feature is to point out is that 4 milligram dose cohort tend to be a little bit more severe in several parameters than those of the 1 and 2 milligram dose cohorts. But in general, it is fairly even matched cohort between placebo and treatment. And also it's a predominantly female cohort, which has been seen in other studies as well. So this is a slide talking about the safety and tolerability. AOC 1001 demonstrated generally a favorable safety and tolerability profile. Most treatment emergent adverse events were mild or moderate. These include procedural pain, anemia. The anemia event were monitorable and reversible. There were 2 serious adverse events, one considered to be related to the AOC 1001 4 milligram per kilogram cohort, which result in the partial clinical hold, as I discussed earlier, and another SAE, which is considered unrelated to the treatment. In the MARINA study, this is the pharmacodynamics, or the what the drug does to the target. So in the MARINA study, AOC 1001 treatment lead to DMPK reduction, as shown on the left, and also splicing improvement on the right. So on the left, you can see that the DMPK change from baseline, which is shown on the y-axis. Each bar is an individual participant. You can see all participants that we have biopsy for show a decrease in DMPK level from baseline. This leads to an overall decrease across all cohort of 42%. On the right, you can see splicing data based on the 22 gene panel composite score that's pioneered by Dr. Charles Thornton. The majority of patients at 2 and 4 milligrams show improvement in splicing as well. Now, for the rest of the talk, I'd like to show the clinical functional and function and strength data that we have gathered in the MARINA trial. In this 6-month trial, we're really not expecting to see a lot of clinical improvement. So these, a lot of these findings were really quite impressive for our team. So on this slide, we're focusing on myotonia. So we're using the method of video hand opening time, and then subsequently rated by blinded raters who are not involved in the trial otherwise. As you know, myotonia is a hallmark of DM1, and where relaxation of key muscle groups is impaired. Here on the right, you can see the difference between the different cohorts, with the gray being placebo, teal color being 2 milligram per kilogram, purple being 4 milligram per kilogram. And this is a comparison between 2 and 4 milligram because both of them got 3 doses in 6 months. The dosing is marked by on the bottom with red arrow. Their dose on day 1 and then 6 weeks after, and then again 6 weeks after. So in first 3 months, they receive 3 doses and then follow for 3 months. So in this graph, it shows that at 4 milligram, the first after 1 dose, the myotonia is decreased by about 3 seconds, and then 6 weeks later is sustained at with it going down a little bit further, and that this is then further sustained through to day 183, which is 6 months. The 2 milligram also show improvement, but the improvement is less dramatic, and also it revert back to baseline by 6 months. This is a video that shows the a person who received the 4 milligram per kilogram AOC 1001. So I want to first orient you in the 2 video panel. The left is the baseline, so it's pre-dose. The right is day 183, so this is a 6-month myotonia hand opening time recording. So that's after 3 doses. And these 2 videos were synchronized so that the the command to open the hand is aligned so that you can see the change in time. So I'm going to repeat after the command just in case the video does not play sound, because sometimes it doesn't, sometimes it does. So here we go. Ready? And squeeze, 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 and open. So this kind of videos are things we have seen over and over again. It's really quite impressive, and really it's consistent with really a lot of the professors have told us that they would expect that myotonia would be improving earlier than any other endpoints. How do I go to the next slide? Sorry, hold on. Here we go. Okay. So the next 2 slides are other clinical endpoints that we have collected during the trial. In this slide, we're looking at quantitative muscle testing. This is a composite of 6 muscle groups from both upper and lower body. As you can see, so this is the strength of the muscle that's collected is then normalized by percent predicted normal to normalize to average healthy population. And going upwards means improvement on the graph. And again, the 4 milligram is performed better than 2 milligram, which performed better than placebo, especially at the 6-month time point. In the next slide, we have the 10-meter walk-run test. So we also see improvement in this assessment as well. Again, the 4 milligram is showing improvement over the course of study versus placebo and the 2 milligram cohort. So we're very excited about this data, and certainly really didn't expect to see the effect so quickly in participants. And then the next, this slide shows the timed up and go. And again, we see similar trend with 4 milligram performing better than 2, which is better than placebo. You see, again, just like video hand opening time, the myotonia, the 2 milligram has a reversal back to baseline after 3 months from the last dose. So in summary, we have shown that we're able to successfully deliver AOC1001 to muscle. And this is the first time I think it really has been demonstrated with this platform. And we have shown DMPK knockdown, demonstrating target engagement. And we also have shown that the clinical assessment showed that there's a potential for disease modification and functional improvement across the multiple endpoints. And this is following the improvement in splicing. So finally, these data have enabled us to select a dose range of 2 to 4 milligrams per kilogram for planning of our pivotal study. So what is the next step from here? So we remain steadfast in our commitment to advance meaningful therapies for the DM1 community. And we want to reinforce our gratitude for the study participants, their family, advocacy partners, as well as the study investigators and their team. Marina open label extension trial is ongoing in participants who were previously enrolled in the Marina trial, and we continue to dose participants in this trial to evaluate the long-term safety and tolerability of AOC1001. We plan to share a first look at the data for the open label extension toward the end of this year. And the results so far really reinforce our belief that there's potential in AOC1001 to disease modify for people who live with DM1. And so we're continuing to partner with the DM1 patient advocacy community in hope to give you more information and also on the next step for our trials, for our program as soon as possible. And in closing, I'd like to share this photo of our team at Avidity that who are committed. This was just taken actually in our collaboration week earlier this month. And we're committed to improve the lives of individuals and families impacted by the rare muscle disease, including DM1. And we're an extremely passionate group and love the work we do at Avidity. And it's our privilege to serve this amazing patient and caregiver and advocacy community. And we're here to answer your questions, and you can reach us by the email on the bottom, patients@aviditybio.com. So that concludes my presentation. Thank you.
Thank you so much. Really impressive talk. Just one question also, the children, what about children for the next step?
Yes, we are definitely looking to the pediatric population to understand how to best leverage our drug for the pediatric community. We do have more work to do, including establishing long-term safety and efficacy for patients in the adults, and we will be evaluating the pediatric population in future trials.
Okay. And just one small question about your slide number 19 about the myotonia. Looking to the view graph, it appears that even for placebo people, the myotonia is a bit improved.
Yeah, so myotonia is kind of a I mean, I think for the myotonia, it's the improvement from placebo is pretty mild, really. I'm looking at 0.3 seconds compared to the 4 second we're seeing with the 4 milligram per kilogram cohort. So I think in a small trial, that's 6 months, which is pretty short term. It's we do see a lot of placebo effects, especially in the earlier time point during collection. And the randomization of 3 to 1, where 3 patients get AOC1001 and 1 versus 1 getting placebo, also contribute to that placebo effect. So we think that really explains some of the effects that we're seeing. In addition, myotonia is quite variable, as you know, and from day to day, there's changes within patients, and also it's influenced by external factors such as, temperature and fatigue and such. So I think we have to take we have to take the data with a grain of salt, I think.
Okay, I understand. Giovanni?
Yes, I want just to ask you about myotonia, because you said that the myotonia is really changing Also, depending also from the diet, if you have an increase of potassium in the food, you have an increase of myotonia. My question is, what about the warm-up in myotonia? Warm-up, because in myotonia,
Yeah. Thank you for the question. The warm-up is definitely a phenomenon that affects people's myotonia, and I think patients compensate actually by warm-up. So for these assessments in the MARINA trial, we asked the patient to rest for amount of time between each of the myotonia assessment, and that's true for the entire trial, and we'll continue to do so for the next trial as well.
Okay. Any other question? Okay, in that case, thank you again, and we will move on to PepGen.
Thank you.
Perfect. Let me just pull up my slides here. Brilliant. Thanks, Alan. So thank you very much for the opportunity to give a little bit of insight into PepGen's program and how we've been developing our program for myotonic dystrophy type 1. So this is the third year in a row now that PepGen have been presenting, and it's great to be able to be back again and to continue to build on the story. So today, I'm going to give a little bit of an introduction into our non-clinical data for our compound PGN-EDO-DM1 that we're working in developing for myotonic dystrophy type 1. So we are a public company, so here are disclaimers. And just as a bit of an introduction to PepGen, so a lot of you are probably already familiar with this, but just as a quick recap and an introduction for those that aren't aware of us. So we were founded in 2018, and our headquarters are in Boston, Massachusetts. We are really, at the moment, focused on rare diseases like Duchenne muscular dystrophy and myotonic dystrophy type 1. And we're at different stages of development for our different compounds. At the moment, we've got about 50 employees on the team, but we're also complemented by a variety of other people that are also feeding into the development of our program, including our scientific advisory board and the community and the patient advocacy boards that we also have as well. And really what we're doing is our mission is to create and to develop enhanced delivery oligonucleotides, or EDOs, for the treatment for Duchenne muscular dystrophy and myotonic dystrophy type 1. So we've heard lots of different opportunities for development for different drug candidates that are ongoing today as well. And again, all of looking at them from the oligonucleotide side of things. And again, we are also using a very similar approach, but with a different delivery modality. So at PepGen, our technology is designed to address one of the root causes for antisense oligonucleotides. And one of the root challenges for that is the delivery side of things. So with antisense oligonucleotides, or oligos, one of the great benefits of being able to use these is that they're able to address the root cause of the disease. However, as I say, one of the big challenges is the delivery side of things. And unconjugated oligonucleotides are not readily taken up into muscle and not efficiently taken into the cells and to the nucleus for the site of action. However, over the last number of years, we've been trying to curate our EDO, or enhanced delivery technology, so that we can overcome this challenge, and we believe that we've been able to find a solution for it. We've been able to show that with our EDO platform, that we've been able to engineer it to optimize tissue penetration, cellular uptake, and nuclear delivery. And we've been able to show that with our platform, we've been able to get good levels of delivery both into smooth muscle, skeletal muscle, cardiac muscle, and at higher concentrations into the CNS as well. But what is our EDO platform? So primarily it's composed of two primary parts. First is our cell-penetrating peptide, which is the proprietary part of our technology. So our cell-penetrating technology has been developed over several years, as I say, really to fine-tune the activity side of things, but also the tolerability and the safety side. And we've been able to show that with our peptides, that we can have this enhanced delivery with our therapeutic candidates. We, as I say, conjugate this to therapeutic oligonucleotides. And for the example for myotonic dystrophy type 1, we're using and applying this to the root cause being the repeat expansion. And with this, then, when we combine our peptide to our therapeutic oligo, we have our enhanced delivery oligonucleotides, or our EDOs. Our technology is scalable, and we've been able to apply it to a variety of different indications and applications as well. So for example, for Duchenne muscular dystrophy, we've got 4 different programs that are at different stages of development, all looking at different subpopulations for individuals that are amenable to different types of exon skipping, including exon 51, 53, 45, and 44. But for today, I'm going to be focusing on our EDO-DM1 program, which is at the later stages of preclinical development. So our EDO-DM1 compound is designed to liberate MBNL1 without reducing DMPK levels. And I know already we've got some really beautiful introductions into the pathology of DM1, but just as a quick recap and a reminder as well. So in myotonic dystrophy type 1, there's a trinucleotide or a CUG repeat expansion in the DMPK transcript. And these repeat expansions create these hairpin loop structures. And these hairpin loop structures bind RNA binding proteins like MBNL1. And these proteins like MBNL1 are very important for the splice regulation of different transcripts. And these structures create a hairpin loop structure that are manifesting in form of foci. And when the MBNL1 protein is bound to it, it's not able to carry out its normal job and this gives rise to the multiple different splicing profiles that are seen in DM1. However, with our EDO-DM1 compound, we have it designed so that it binds to these CUG repeats, and then it liberates MBNL1 from these pathogenic foci. And by liberating of MBNL1, we can then restore the physiological splicing, which can then improve the phenotype of DM1. And again, we've seen today that there's different ways of applying antisense oligonucleotides for a DM1 perspective. And for ours, as I say, it's liberating MBNL1, but again, it's designed to not degrade the DMPK transcripts. So with the missplicing that occurs in myotonic dystrophy type 1, it really impacts a lot of different systems within the body, including the respiratory side, the GI, the cognitive, the cardiovascular, and also smooth and skeletal muscle, which creates a variety of different manifestations of the disease as well. And as I say, with our EDO platform, we've been able to do a lot of basic research and understanding the platform from a broader perspective. And we've been able to see that we have great delivery to a lot of the critically impacted systems impacted for myotonic dystrophy type 1. As a brief introduction, we've done a lot of research over the last number of years to really explore the benefits of our technology and applying it to DM1. And from the pharmacology side, which is what I'm really going to focus on today, we've been able to look at it from different models. So first, we've been able to look at it in our DM1 patient-derived cells. Then we've also looked at it in animal models, including the HSA-LR mouse model as well. But we've got a myriad of other studies that we've done that I won't go into today, but they've also helped to enable the further development and progression of our program as well. So starting at the very beginning, we began to look at it from the in vitro perspective. So looking at it in DM1 patient-derived cells is critically important from a proof of concept side of things, and also to understand from the mechanistic, but also the impact that we could potentially have for correcting splicing. So for this, we've looked at cells from an individual with myotonic dystrophy type 1, and this individual has about 2,600 repeats. So as I mentioned earlier, one of the hallmarks for DM1 at the cellular level is these kind of pathogenic foci that we can see here as the red dots, or if maybe you're not able to see because of the size of the screen as well, we've got arrows pointing to them as well. So these foci are the repeat expansions that are present in DM1. But the MBNL1 protein, which, as I mentioned, is critically important for normal splicing, that also co-localizes with these repeat expansion foci. And that means then it's not able to carry out its normal function. So first we wanted to see, are we able to impact foci levels, but also are we able to look at liberation or release of MBNL1 from this repeat expansion? When we do a single administration of our compound in vitro in these differentiated myotubes, so myoblasts, we're able to see that at constant, we've got a dose level response in the reduction of the number of foci per nuclei. And again, at higher concentrations, we're able to see a reduction of 54% of foci. But importantly, we're also able to see that in the cells as well, we're able to liberate MBNL1 from these foci as well. And as you can see in both the images from the foci perspective and from the MBNL1, we've got a huge reduction in the amount of foci and the bound MBNL1 following treatment with PGN-EDODM1. And while it's great that we're able to see that we've got this benefit, really understanding, can we have a mechanistic impact, which is more important. And to look at this, we did a splicing evaluation for this. And again, we were able to look at a variety of different transcripts and do a composite analysis for this. And we can see that, again, we've got a beautiful dose-response relationship when we treat with our EDODM1 compound, and we've got up to 69% correction across a variety of different transcripts as well. And while it's great that we're able to see this, part of the technology that we're developing as well does not degrade DMPK transcripts. So we've done some evaluations to see what does that look like, both in the same DM1 patient-derived cells in vitro, but also in non-human primates as well. So first, we looked at the DMPK levels in the patient-derived cells, and even at higher concentrations of 20 micromolar, where we're seeing good levels of foci reduction, liberation of MBNL, and splice correction, we're not seeing any degradation or decrease in DMPK levels. But again, we also wanted to see that it's great that we can see this in vitro, but ultimately we're going to be going into people, but in the interim in between that, we do a lot of preclinical evaluations in mice and in non-human primates. So looking at non-human primates, we did administrations every 2 weeks for 3 doses. And even at concentrations of 60 mg per kg, we did not see any changes in DMPK levels. But really, we want to see what is the activity levels looking like in vivo systems where the repeat expansion is present. So bringing it forward beyond the in vitro side of things and bringing it into the in vivo. And I think some of the other colleagues on the phone today, they've already introduced some of the mouse models that can be used. But just as a quick recap, so the HSA-LR mouse model was first developed in 2000 by Charles Thornton and his team. And this really is one of the widely used, most routinely used mouse model for myotonic dystrophy evaluations. So this is a transgenic mouse model that has the CTG repeats in a human skeletal actin transgene that's been inserted into the mouse. So in these mouse models, they have about 220 to 250 repeats. But what's really beneficial about this mouse model is that we're able to see that with the skeletal muscle specific phenotype, that there's some of the DM1 abnormalities are also present and recapitulated in this mouse model, and we're able to see how our compound can potentially improve or impact these as well. So similar to the side of things that we could see in the cellular side of things, we can see that there's the presence of foci in these mice. We can also see that MBNL1 is sequestered to these foci, and then also it gives rise to downstream missplicing events at the molecular level, but also at the physiological level, we're also able to evaluate myotonia. So with this mouse model, just to give a little bit of an insight into it, when it's resting and also in normal mice, the hind limb muscles are tucked under the mice. However, when you want to kind of physically observe what the myotonia looks like, the mice have these splayed legs behind them as well. And this is the typical phenotype of what it looks like when the myotonia is present in these mice after excitation. So we wanted to evaluate our compound after a single dose in this mouse model. So we administered different concentrations of our EDODM1 compound and evaluated what the results look like 2 weeks post-dose. So first, we looked at the molecular level, looking at the missplicing evaluation. And as I showed for the in vitro work, we did evaluation across multiple different transcripts, but here I'm just going to show you 2 of the multiples that we evaluated in vivo. So first looking at ATP2A1 and then also looking at CLCN1. And we can see that with a single administration that we can improve the profiles in the HSA mouse model to look like control mice. So after a single 30 mg per kg administration, we're able to see that we've got 91% correction of ATP2A1, which is a transcript that's critically involved or associated with muscle excitation. In addition, we've also looked at CLCN1, and again, after a single administration at 30 mg/kg, we're getting close to complete correction and restoration to wild-type levels for CLCN1. And again, this is another critically impacted transcript that's evolved with muscle excitation. But more importantly, the CLCN1 profile is connected with the myotonic profile. And we were able to evaluate myotonia in this mouse model using a quantitative evaluation. And we're able to see after your single administration again that we've got correction of myotonia, which is, as I say, connected and providing the connection through from the missplicing and also correcting it at the physiological level as well. But while it's always great to see what this looks like from a graphical representation, I think for me, always being able to visualize it in either a photo or in a video is most beneficial. So as I said earlier, we've got different, when you pinch the hind limb muscles of the HSA mouse model, you can see the hind limb muscles kind of staying contracted behind them and they drag along. However, when we treat with the EDO-DM1 compound, when you pinch the hind limb muscles to try get that myotonic discharge, we don't see that any longer. So those hind limb muscles are staying underneath the mice and they're behaving as what a wild type mouse would look like as well. So again, looking and seeing that we're able to correct both in vitro, but also in vivo after a single administration is incredibly promising. But during the drug development process, we also want to see, is there any way that we can help understand the dosing frequency as well? So we wanted to take this evaluation and take it a step further and looking at what the duration of effect or how long does this correction persist for. And for this, we continued on with the single dose administration side of things, and we wanted to look beyond 2 weeks. So looking at 12 weeks and 24 weeks post a single dose. So we've done this evaluation in 2 different skeletal muscles, both the quadriceps and the gastrocnemius. And we've been able to see that we've got sustained correction of the 2 transcripts that I showed earlier for the HSA mouse, not just at 2 weeks post-dose, but also 12 and 24 weeks post-dose as well. And the benefit of having this long duration of activity is hopefully that we'll be able to have that translate into the clinic and will result in less frequent dosing as well, which means that potentially less visits for the dosing. So as a quick recap for the data that we've presented today, we've been able to show that we've got reduction in toxic foci, liberation of MBNL1 from patient-derived cells. We've also got correction in missplicing in multiple different preclinical models, those with long repeats, but also those with short repeats as well. And for our DM1 mouse model, we were able to see robust missplice correction and reversal of myotonia following a single dose up to 24 weeks. And again, while other technologies are designed to degrade DMPK, we've been able to see in different preclinical models that we're not degrading DMPK, but still having high levels of activity. And just to touch very briefly on the toxicology side of things, we've been able to see that our compound has been well tolerated through a single 90 mg/kg dose in non-human primates as well. And again, this is just a snapshot of some of the preclinical development for our EDO-DM1 compound, but across the platform from our EDO technology as well, we've been able to show that we have delivery of pharmacologically active levels of oligonucleotides in humans, and that's coming from our EDO-51 program for Duchenne muscular dystrophy. But also we've been able to show that we've got delivery of oligonucleotides to the CNS in non-human primates, which could again be very beneficial for DM1. So this teamwork has been part of a bigger group organization as well. So we've done a huge amount of work internally, but a lot of the work is also being led by our incredible collaborators. And I think Denis is also on the call. So a big thank you to Denis and to Arnaud for their work on this project as well. But most importantly, thank you to our community and to our advisors for their advice and support throughout this program and for the development as well. So I'll pause there and happy to take any questions.
Thank you so much. Well, we have a question, of course. At the beginning of this year, you have announced clinical trials for this year. Then we learned that FDA placed a hold on this trial and would give you reasons for that within 1 month from April. Did you get the information and why is it in hold?
Yeah, absolutely. So at PepGen, obviously with the promising preclinical data that we have, we ultimately want to bring that into clinical trials as quick as possible. Again, we're very much a thoughtful and considered team and want to make sure that we're developing compounds that are safe and bringing them to the community as quick as possible. You're right, we did get placed on clinical holds and we just received that letter and announced that at the end of last month. And this is something that we're working very closely with the FDA to lift the clinical hold as quick as possible. But at the moment, we're not able to go into much more detail than that, I'm afraid. But we are hoping to lift it as quick as possible so that we can continue on for a phase 1 clinical trial.
Does it mean that we can still announce that the clinical trial is on its way?
Well, we need to get off clinical hold, but absolutely, once we can lift that, we will be trying to bring it through as quick as possible. Yes.
Any question? No question at all. It was so clear. Well, if there is no question, I think it's time to finish and wrap up the meeting. Thank you, everybody, for this very exciting meeting. If we go back 2 years ago, trials were announced, studies were ongoing, but it was in the future. And now we can clearly see that future has come to present, and trials are very in the way to start very soon or already on active. That means very exciting for patients, and we will keep in touch for that and be sure that we will stay with you to help and assist and accelerate as soon as we can do. Okay, any other conclusions from the meeting? Katie, you want to say something, but you're on your mute.
I'm so glad to be here after 30 minutes.
Okay, so I am very disappointed about that. Really, it was not, I don't know what happened, but Teams was really down for a long time in this afternoon in most of the countries. Some of you did succeed to connect, some of you did not. And we apologize for that. And we will have to do something to fix and to maybe to do a second time for this meeting later on.
So-
We'll discuss that in the board.
Yeah. It is recorded, so
Yeah,
we can see it again for the time that will be best. Okay.
Okay, one of our representatives will edit the file. Will cancel inappropriate scratches and some other stuff, and we'll put on YouTube channel of Euro-Dyma the movie of our meeting. I hope it will be okay, and I think it will. Thank you again for your participation, and see you. Maybe Anne-Berit, you want to say something?
I think it's promising to hear that some of the pharmaceutical companies are planning for including children with DM1 in clinical trials. That's very good to hear. And also the last one that talking about penetration to the central nervous system, because that's delivering, because we know that central nervous symptoms are quite devastating and needs to be addressed. So not just the muscles, but also the CNS. So that's a challenge. So yeah.
Yeah, Giovanni, any comment? With the microphone on, please.
I agree completely. First of all, I want to thank all the speakers who are very good, very clear. Also, I had also problems to connect. Well, but you know what happened. Well, doesn't matter. But nevertheless, I agree completely to Anne-Berit, my good friend Anna. Hello, Anna. That the big challenge will be the brain, because this is a big point. We know that mainly for the congenital form, for the childhood form, but nevertheless, now we have an avenue, we have a pathway, so we should move on, you know. And I hope that really we are very close to the cure and to the treatment. Okay, we cross fingers. That's all right.
Thank you so much, Giovanni. Excellent.
Okay.
Basil, the chairman of the scientific board, maybe you want to add something?
I think it is an excellent overview on what's going on in the field, and I can only repeat what was said before. So nothing novel to add. So I'm curiously looking forward to the future and the IMC 14 meeting next year in Nijmegen. And hopefully a lot of the presentations of today will then be given with even more details, hopefully.
Okay, thank you so much, everybody, and see you next. Bye-bye.
Okay,
Thank
bye-bye.
you.
Bye-bye.
Bye.
Thank
Bye-bye.
you.
Bye-bye.
Thanks, everyone.
Bye.
Thank you. Goodbye.
Bye-bye.